Hi, it's Matt. Today I'm trying to understand the meaning of life. And by that I mean how to sequence DNA. I'm at the Francis Crick Institute, one of Europe's largest biomedical research centres, whose genetics department found one of the major causes of inflammatory bowel disease just this month. And they're going to be teaching me how to use a big robot to chop up human genes. That sounds a little scary. Ooh. As if this actually exists. What? That does it all itself?
Yeah.
This looks like science in films rather than science in real life. Sorry, I have just spotted Buffy the Vampire Slayer and Angel there. The lab I'm working in today has a really cool function here at the Francis Crick Institute. I get to wear a lab coat for this one, because I'm going in a lab. Scientists send DNA samples they've collected here, then the team chop it all up and prepare it for analysis.
Hello.
I'm Matt, nice to meet you.
Nice to meet you. I'm Maria.
I'm Ashley.
Hello.
Nice to meet you.
As they so eloquently said, this is Maria, and this is Ashley. Maria runs the sequencing lab at the Crick, and Ashley is a laboratory research scientist. So, from what I can remember from school 20 years ago, DNA is the recipe for animals, humans, life.
Yes.
And it's the recipe for how the cells are made. Is that right?
Yes, that's it. Yeah, it's the instruction manual of your cells, for example. Yeah.
That is all I know. Please tell me more.
Yeah. So all of your cells in your body contain DNA, and they all contain the same DNA. But then you have different cell types. And how do you think this can happen?
For those of you who needed the same refresher I did and would prefer a visual aid, here's the general idea. You probably all recognise this, the spiral shape we all know of as DNA, which, when unravelled, looks like a ladder. The rungs of this ladder are made up of different chemicals which come in pairs. There are 4 different types of chemicals. We represent them as the letters A, T, C and G. A pairs with T and C pairs with G. So one side of our DNA is basically just the inverse of the other. Therefore, if you split the DNA in half, you can still read the DNA sequence.
So not all of the bits of the DNA are active in all of your cells. It's only a few of them.
Right.
And depending on which one is expressed is the RNA, is the active form of the DNA that will determine which cell type you have.
Maria said something there, RNA. RNA is the active form of your DNA. Unlike whole DNA, RNA is a single strand. If DNA is the blueprint for the cells, RNA is a thing that carries out the instructions. So DNA makes the RNA, and the RNA makes the amino acids that make you.
Sequencing basically is reading the order in which the letters are arranged. So imagine that the instruction manual, if you have a book, is written in the 20-something letters depending on your alphabet. So we have a 4-letter alphabet in the DNA, A, C, T, and G. And sequencing is finding the right order in which these letters are arranged, and then they compose your instructions.
I know DNA sequencing is important. It's a phrase I've heard before. But what do you do? Like, once you've got that recipe all written out in a line, now what? What can you use that for? How do the researchers use that information?
So we have loads of projects. For example, we have the flu project. So we have the World Influenza Center downstairs, and every week they bring samples from different places in the world, and we sequence which strains of flu are happening in the different places, so then they can design the vaccine.
Yep, you heard that right. A big part of what Maria's lab does isn't sequencing human DNA, but virus DNA. In fact, the lab was instrumental in the early days of the COVID pandemic. Those PCR tests you may have taken? Well, if your result was positive, then your spit was being sequenced in a lab just like this. They can even use sequencing to treat cancer.
You can take the DNA from the cells that are healthy and the cells that are tumorous. And usually what happens is that cells have rearrangements or they have mutations, and you can find which mutations are causing the tumor, and then use drugs that will attack only the tumor cells rather than your normal cells.
Because the DNA is the recipe and something's gone wrong because the recipe's changed, so you're comparing the recipes.
Exactly.
So what am I going to be doing first?
DNA is really long, so if you take the DNA from one human cell and you stretch it, it's more than 2 meters long. And we have to chop it up to be able to sequence and read it to smaller pieces, and add some adapters to be able to read it. So to do that, we have some machines that help us.
I'm going to be guiding you through our lovely robot we've got over here.
That's one hell of a robot.
If you'd like to come with me.
Okay, so now we know what DNA is, it's time to see this process in action. First, I'll be learning how to prepare DNA for sequencing, and then I'll be seeing how they actually sequence the DNA.
Most of the samples we get sent, they have to be prepared before they can be sequenced.
And what does prepared mean?
So they essentially have to be made readable by the sequencer. It essentially just reads your DNA. Before we can do that, we need to add little adapters so they can actually go onto the sequencer.
Very broadly, this process of adding little adapters goes like this. You take your sample. Actually, in this lab, they take 96 samples at the same time on one metal plate. Now that's efficiency. And they add certain chemicals, or adapters, to it. You then have to heat it up to a very specific temperature and spin it in a centrifuge to make sure all of the sample is collected together. At the end of the process, our DNA has gone from being a long coil to a bunch of shorter segments, each with a little adapter on the end. And in this lab, the whole thing is done by a super cool robot. Luckily for me, the person showing me the process has a lot of experience.
So I've done a lot of work with COVID samples.
Right.
So this institute, it has tested over 750,000 samples for
Wow.
COVID.
So the chances are that my spit has been through this room.
Maybe, yeah. Who knows?
When I've done a PCR test, it's come through here.
Possibly.
Yeah.
For every sample which was a positive sample,
Yeah.
we then sequence the virus, so that can be used for further research.
Right, so they do the test. The test works out that this has some COVID in it, and then they do the DNA analysis of the COVID. But for them to do that analysis, you need to have just made the stuff happen so then they can do the research?
We need to read all of the sequence which makes up the virus.
Got it. So you are the cogs that make the research happen.
Yeah, it's not as simple as like on telly where they put a sample on a sequencer and it pops up with this is your DNA. There's a lot more work which goes into it.
This is the kind of thing I always wonder about. Like, they say, oh yeah, we just sequence it. But it's not just sequencing it. You have to sequence it and prepare it beforehand.
There's always a team of people behind it.
So let's see the preparation then.
Yeah, so if you'd just like to click resume.
Okay.
So most of the process used to be done by hand.
Yeah.
We now have robots which can do the whole thing for us. Shortly, you'll see this robot arm take the plate from the liquid handler. The purpose of that is adding parts of the recipe, parts of the ingredients to make the samples readable.
After the arm has picked up the plate, it's onto the first stop, the plate sealer.
So you have to put a very firm seal because some of the temperature it reaches can be up to 96 degrees. So if it's not sealed, you'll get a lot of evaporation.
Once it's been sealed, the arm collects it and moves it down to the transport layer to take it to the other side. Oh, there's something moving under
Yeah,
there.
this is...
Oh, is that magnets?
It's a really cool piece of kit. So each one of these is separate. It's got a little magnetic arm underneath, which will move the transport layer across.
As if this actually exists.
Yeah.
This looks like science in films rather than science in real life.
No, it does genuinely happen. It's quite amazing.
So that's another robot arm. So that's
another robot arm. You'll see
drag
it, pick it up, and then it will go down to a little instrument down there in the corner. This changes the temperature very rapidly to get the enzymes, the ingredients, to their optimal working temperatures.
Should we go around there to see it?
Yeah, absolutely. As I previously mentioned, some of the temperatures can get very high, so you'll get some evaporation.
Yeah.
This can then condense on the side of the wells.
Right.
So once that's finished, it'll go into that big red box.
Yeah.
Which is a centrifuge. So it will spin it really fast.
Yeah.
Getting all the liquid back down into the bottom of the wells.
The thing that strikes me about this is that it isn't just the robot machine. You've got a robot arm, but you've got all of these separate machines which appear to all be made by different companies, yet it is all integrated in such a way that the arm can move all of the things together.
It's quite on the cusp of what we can do at the moment.
And I suspect that you have spent years studying, and it's way more economical, both time and money, I suppose, for the institute-wise, to use your intellect rather than just manually moving things. I see this machine's called Hamilton.
Yes, it is.
Does it sing in the musical as well, or?
It doesn't. You can program it to play a little tune once it's finished.
Really?
I know. It's something we haven't done yet. Ooh. Yeah, so
cool.
that's just its way of saying that it's finished, it's completed.
So it does all the stuff and it plonks it back there ready for the next stage of the process.
ready for the next step.
In the course of making this series, I've got to work with so many people who are outstanding in their field, and that includes the team working behind the camera too. And while I choose to release these videos completely for free, I'll be honest, they do cost several grand to make, and I haven't got an advert for this one. So if you'd like to join the group of wonderful people who are supporting me on Patreon, you'll get to see some bits from my day that didn't quite make it in this video. The link's in the description below, and I'll be honest, you can join for free too. Most of the stuff I put on there isn't paywalled, so come along, join the fun. With the sample prepared, it's time to move on to the actual sequencing. So for this, Maria took me to another lab with some even more expensive toys.
So we have these ones here on this side of the room are the Illumina type sequencers.
Okay.
So this is sequencing by synthesis. So
Okay.
basically, you've chopped your DNA into little pieces, you added the adapters on the end,
Yeah.
and then we have to put them into the machines.
Okay.
So these are what we call flow cells, which are basically glass slides that contain little sequences of DNA all
Okay.
over the glass slide. And then we put it into these machines.
Yeah.
And then we add the sample that you just prepared, and then the machine flows the liquid through it. And the DNA, because it has these adapters at the end, it will bind to it, and then it will allow the DNA to be attached and not go away.
Right.
So then there will be millions of molecules of DNA here. So for these baby machines, the Mi6, we can sequence up to 25 million different bits of DNA.
It's the baby machine, but
This
it
is
can
the baby,
sequence
25 million.
25
Up
million.
to 25 million. What we do is we add the different nucleotides, so the letters
Yeah.
will have different colors, so red, green, blue.
Okay.
And so we add the 4 at a time, but only one is added to each molecule at a
Right.
time. And then we take a picture. Because they have the colors, the camera will take the picture.
And do you mean that literally? You are actually putting colours on that and taking an actual photograph?
Yeah. So we take a photograph.
So you can actually see it?
Yes, so you can
Wow.
actually see it. Then we remove the colors, we add a new batch of colors, and so we add one nucleotide at a time, and we can read the sequence like
So
that.
you'll take the blue one, then you'll take the green one or
Yeah,
whatever.
so
and
you
then-
take the pictures, and then you compose your sequence by compiling all of your images and saying, okay, this was first blue, then red, then green, then yellow, blue, green, so it's ACTG, whatever. And these are the baby machines.
Yeah.
So we use these every week for sequencing flu. As I told you, we have
Yeah.
the Influenza World Center here. And you can do smaller genomes here. But for things like the human genome, like
Yeah.
when you have cancer patients or something, what we do is we need higher power. So we have these other machines, like this one is the NovaSeq. It allows you to sequence up to 15 human genomes at a time in less
Right.
than 2 days. And this
Wow.
is it's the same principle. It's a glass slide, but this one contains many more small Lego bricks to bind your DNA to
And
it.
is this one bigger because you're using longer strands?
No, it's not for
It's
longer
just because it can
strand.
do more at once?
It can do more at once.
Right.
So basically, these machines have cameras, and you just have to take more pictures.
Wow.
It can do many more. So 9 billion versus 25 million. You can
Sorry,
see
I have just spotted Buffy the Vampire Slayer and Angel there.
Yes.
Is that is this machine called Buffy?
We call this one specifically Buffy and Angel. So each side, it has two sides.
Oh, you've got two sides,
So
yeah.
each side can do one flow cell at a time. And because it's called side A and side B,
Yeah.
so side A is Angel and side B is Buffy.
Oh, yeah,
So we can tell if somebody fails, oh, Buffy failed this time, or we have only Angel running today. So in this case, you see that there is green, so
Yeah.
only Angel is running today. So now I'm going to show you the other technology
Yeah.
that we are using in the facility, and this is Oxford Nanopore technology.
Okay.
So these machines are massive.
Yeah.
You can't take them with you when you're
No,
doing research.
it's not going to fit in the back of your car.
No, but we have small sequencers that you can actually put in your pocket.
What? That does it all itself?
Yeah, it's a different technology. So it's called Oxford Nanopore.
Yeah.
And basically, it doesn't have a glass slide.
Yeah.
It's a membrane that contains pores that have proteins,
Yeah.
protein pores. And when the DNA goes through it,
Yeah.
it causes a change in voltage. And the change in voltage is because every base is a different composition of molecules. So
Right.
it has the different composition. So then when it goes through the pore
Yeah.
changes the voltage, and then it's read by the machine. So it's it's kind of like a semiconductor
Yeah.
style.
Oh, that's really cool.
And what is really cool is because that one was taking images, you need to wait until all the images are taken to be able to read the sequence.
Yeah.
Whereas this one, as it goes through, you're reading the letter. So you can see what
You
you're
get the
sequencing.
direct information out of it.
Exactly. So then what you can do is you can take this with you and imagine that there is an Ebola outbreak in Africa and
Yeah.
you want to see, is it Ebola or is it Marburg? Which type of virus is it?
And you don't need to take all the samples with you. You can
don't
just
need to,
do it.
Yeah, and you don't need to take a 1 ton sequencer with you. You can take this machine and do it in the field. Take
So
it
if
with
that
you.
can do 25 million at once, what's the number on that?
This one is
going to be
different.
much smaller,
yeah,
isn't it?
this is smaller, and also it's different because this machine also does longer reads. So
okay.
these ones do up to 500 base pairs. The baby one does up
Yeah.
to 600 base pairs. The big ones that do up to 150 on its end.
Yeah.
This one can do really long molecules.
Yeah.
And this is really good for checking virus or assembling new
Okay, yeah, yeah.
genomes, etc. It's a new technology. It's different.
So it's similar in that it's both sequencing, but this is just doing it in its own new way.
We were using this in the early days of the pandemic to sequence really fast the viral strains to see if it was Omicron or Delta. These ones allowed you to do this really fast. But when you want to do human genomes, you need to go through the larger machines, so the
that's cool.
PromethION and the GridION.
So what does it show you on the computer? Oh, you're
So
showing me that now.
I'll show you, yeah. So basically, as I told you, these are pores where the DNA is going through, and this machine will show you the status of the pores. So basically, each of the squares will be a different pore.
Yeah.
And
Do those colors match the same colors you use on the...
No, the colors mean that the pore is active or not active.
Okay.
So the light green ones is pores that are getting ready to sequence.
Yeah.
The green ones are pores that are actually sequencing, so there's DNA going through. And then the dark blue ones are the ones that are recovering. So after they've sequenced, they need to recover, and then they will get ready to sequence again.
That is so cool and amazing the way that technology can progress from having like small machines that can do a bit, then big machines that can do a lot, and then to have it in your pocket.
Exactly.
So this information then, now we've got it, that will go through to another lab who can use it in their research?
Exactly. So then everything produces text files. So DNA, as I told you, is the instruction of the manual of our bodies, for example. And then we produce a big text file that we will send to bioinformatics service that can analyze it, or to the researcher if they can do their own analysis.
That is so cool. Thank you so much for showing me all of this.
You're welcome.
I could never have known all of this happens in here. Oh, it's amazing.